In this exercise, we will perform an analysis of p53 mutations in all the primary tumors that are involved in the PanCan-12 dataset. And here, we will first perform the general p53 mutation spectrum analysis on all the PanCan-12 tumors to look at what are the common mutations that are associated with all the 12 tumors that we are going to investigate. And next, what we are going to look at, we will be looking specifically on the p73, for the p53, R273, R175, and also NS mutation in the PanCan-12 tumors. Follow up, we will then try to look at the expression of the wild type p53 versus the p53 mutants in terms of the mRNA and expression and also the protein expression, and see if there's any correlation. And finally, what we will do is we will also do the analysis of the clinical outcomes, mainly the survival outcomes in tumors that are associated with p53 mutation. Okay, in the first exercise, we are going to perform the analysis of p53 mutation status or spectrum across all the PanCan-12 tumors. Now, one of the best way to actually get the p53 mutation spectrum is actually using the data set, the same TCGA data set. You can access it from different platform, but one of the easiest way would be to use the CBio portal. Now, to look at the PanCan-12, first you need to understand what are the tumors that are included in the PanCan-12. So this is the list. So you can just Google PanCan-12 and you can find out what it is. So this is basically all the tumors that are included in the PanCan-12 dataset. So now if I go back to the CBio portal, what I have done so far is that basically I have selected all the PanCan-12 tumors here. So the one with a tick. Okay, and what you will notice is that I will try to use a TCGA provisional data set as much as possible, because that is the most updated and the most comprehensive data set at this point. Now, so once you have selected all the tumors, okay, then you need to select the data type priority. Now in this exercise, we are going to just select the only mutation part, and we are not going to look at copy number alteration altogether. Now the reason is that we are just interested in the DNA mutation. So this is important because if you select both the mutation and CNA, the results that come out later on will be reflecting of both the mutation and also copy number. So I will come to that later on. So now the next thing you want to key in would be the gene set. Okay, in this case here, we are just interested in the one gene only, which is p53. So next, I'm going to submit. So and then the results will come out soon. Okay, the first page you will land on would be this overview page. So here they will tell you straight away, okay, what is the alteration frequency, okay, of all the tumors that you have selected. Now, remember the alteration, what is this alteration frequency? Remember just now we select the data set to limit to just looking at DNA mutation. In that case, the alteration here refer to DNA mutation. However, if you select in the previous page that you are looking at DNA mutation and copy number alteration, then the alteration frequency here will be referred to mutation and copy number alteration. So be careful of the data type that you select. Now, in this graph here, what it tells you is basically in ovarian cancer, there are more than 80% of the cases have at least one type of p53 mutation. Okay, so now you can download this graph either as PDF, okay, or you can, if you want, you can download the vector file, which is SVG. So now, if you download PDF, this is much easier for you to manage. If you are not sure how you are going to open the SVG file. So in the separate exercise, okay, I will show you how you can actually convert the SVG into another image file. But in this case, at this point, okay, if you are interested, you can just download the PDF and then you can just open it up, and then you can just, for example, okay, copy and paste the image and put it into the PowerPoint. So the image should be high resolution enough for you to put into the PowerPoint and also for any type of thesis or hard copy printing. Okay, so anyway, come back here. Now, there are a few things that you can actually alter. For example, okay, y-axis. So at the moment, it's looking at frequency. If you want, you can change it to absolute counts as well. So that will give you the number of cases of each tumor. For example, here, head and neck cancer, you have about more than 366 cases that have one type of p53 mutation. Now, also of note is that, well, if you go back to the frequency chart here, is that, okay, look at here, okay, these two cases, for example, AML and also the renal cell carcinoma, they have relatively low mutations. Now, we have to be very careful, okay, because once we stratify all the p53 mutation into R273 or R175, we have to relook again what are the tumors that actually have high enough number for those mutations. If any of the tumors do not harbor any of this mutation, then we will probably exclude it from our analysis. So, okay, so that is the overview tab. Okay, after the overview tab, you also have another tab which is mutations. Now, this information will be quite interesting. It straight away gives you a snapshot of the spectrum of the p53 mutation across all the tumor types that you have selected. For example, okay, you have about 1081 missense mutation, 556 truncating mutation, and so forth. So if you are not sure, okay, what the color code stand for, you can always click on here. It will show you, it will explain to you in well in in a comprehensive format what it is stand for and what it is included. Okay, so you can also customize, okay, your y-axis value, but in this case here, most of the time we won't bother so much about it. And you can also save it as PDF and SVG as you like. Now, if you look at this diagram here, it's basically summarize all the mutations in p53 in different part of the p53 mutation, in different part of the p53 genes. Now, if you are interested in just missense mutation, for example, you can click on this button here, okay, and it will only display the missense mutation and not the truncating and other mutations. Okay, so and if you want to look at truncating mutation, for example, you can okay click here to reset all the filter. But before we move on, okay, I just want to point out, okay, it straight away give you the analysis that the most common hotspot missense mutation, okay, in the pan-KRAS tumors that we are seeing are basically R27, uh, mutation at the R273, and also mutation at the R248, and also mutation at the R175. Okay, so now let's look at the truncating mutation here. Okay, we just reset all the filters, and then when we click on truncating mutations, okay, then you will get another, well, this will be the spectrum for the truncating mutation. Now, it will be good to read up what is meant by missense mutation, inframe mutation, truncating mutation, and so forth to fully understand and comprehend what are the analysis that we have done so far. Okay, before we move on to another type of analysis, so the last tab that I want to show you is this expression tab. If you click on the expression, so it will give you the mRNA expression at this point, basically, of all the tumors that you have selected, okay, in the first page. Basically, here what is show is basically these are the mRNA expression based on RNA-seq data, and then you have different type of mutations and what is the expression that they have. What you can see here is that, well, most of the, for example, nonsense mutation, you will have a much lower, okay, mRNA expression. This is kind of like expected. So again, this will give you a quick overview, okay, in terms of the expression level. Now, by default, it will sort basically by the cancer study type in alphabetical order. Well, if you want, you can sort it by median. That will be easier for you to look at and discuss. For example, you can say that, okay, uh, in terms of what you call it, in terms of p53 mRNA expression, ovarian cancer seems to be the highest compared to, let's say, clear cell renal carcinoma. Okay, so now this information is just okay. Serve as a good reference in terms of conclusion wise. It probably doesn't help so much. I think it would be good to include it into the one of the figure as well, so that, well, you can actually discuss it further. Okay. Okay, once you have finished all the analysis, this is the end of the exercise one. You should be able to get some graph, something look like this, look with the count, okay, with the p53 mutation counts number in terms of cases as well as the frequency, and then you can also stratify the p53 missense mutation and truncating mutation and in-frame mutation to show basically which part of the genes are basically highly mutated. And again, the whole idea to showcase here is basically if we are looking at p53 missense mutation, which most of the time also associated with gain of function mutation. So to justify that, okay, the R273 and also the R175 was indeed one of the hotspot. While we are not looking at R248 at this point here, but at some point it might be interested to look at it as well. Now for truncating mutation, we are not so much interested in it. So basically for truncating mutation, most of the time it's nonsense. So we will assume that most of this truncating mutation would actually give a loss of function as well. And there are quite a number of inflamed mutation. These are the ones that will actually cause the change of the whole protein sequence, and we do not fully know what is the consequences of it, and most likely we won't include this into our analysis. Okay, and finally the last one, the expression, as I just mentioned earlier. So this is the data that good to show and also to describe, but in terms of the analysis, I don't think we can make much of it. So that's the end of exercise one. In the second exercise, what we are going to do is we are going to look specifically into the R273, R175, and also the nonsense mutations in all the PanCan-12 tumors. To identify the cases that are associated with the P73, R273, R175, or NS mutation, so we will again turn into the C bioportal. Okay, so now if you go into C bioportal, I will go back to the home page again. So now if you go to here, now what you would do is you will select individual tumors. Okay, for example, I will start with, let's say, breast cancer here. Okay, use the provision data set. And again, they have a lot of options here. What I'm going to do is I'm just need to check the mutation box here. Now, the second part on the select patient part, okay, make sure you select the sequence tumors. Okay, so if you look at it, so there are quite a number of cases. If you select the all tumors, the likelihood is that, well, you have some of the tumors which are not being sequenced. Now, if they are not being sequenced, then the results will return with not available, well, with the terms not available or NAN. Now, when you see that, okay, because in the CBAR portal data set, what happened is that even the wild type, they will also indicate it as NAN. So if you use the all tumor set and when you see the NAN, you won't be able to know whether it is because the tumor is not being sequenced or it is because it is wild type. Now, to be safe, what I would do is I would just always select this part here, which is the sequence tumors. Okay, and then you put in your p53 genes again. So you put, then you submit it. Now, okay, then you will get to a result page, something like this. Okay, and then if you go to mutations, now this is very useful to give you an overview of the whole spectrum of p53 mutation in breast cancer. If you are interested, you can actually copy this image and then put it into the PowerPoint as well. But from here, what we know is that while the predominant p53 mutation in breast cancer are probably the R175H point mutation, and the second common one will be the R273 point mutation. So now if you scroll down, it will give you all the sample ID matched with the different amino acid changes or the mutation. Okay, so now the problem with this page here is that it only gives you the cases that have p53 mutation. It doesn't give you the sample ID that are wild type p53. Now to get the full data set of the p53 status of all the primary tumors in the TCGA provisional set, what I would do is I will go to the download page here. All right, so you will see it. Okay, well, you can actually download the tab delimited, okay, format, or what I will do here, I will just press this one, the transpose matrix. Okay, so when you press that, it will again download all the data into the notepad. Okay, you just copy it and then you paste it into the Excel format. Okay, so now then you have all the different gene ID with the different p53 mutational status. Now remember, because the tumors that we selected is actually the set that are all sequence tumors, so all the data that return with the terms NAN is actually referred to those that are wild type p53. Of course, you also have some other mutation. For example, here that is ended with an asterisk. Those are mainly the truncated mutation. You have splicing mutation as well, and you also have some of it which have frame shift. For example, like this. This represent the frame shift mutation. So now, so what you would do here is basically, yes, this is the breast cancer data set. I will just label it here, tumor type as BRCA. Okay, so then you can copy it all the way down, all together to the 900 over sample. Okay, and you can save it. Then after that, you can repeat the same process again from different tumors. So I'm going to uncheck this box now and move on to next. Let's say if I'm going to look at, let's say, colorectal cancer, for example. Okay, so I'm going to just select the mutation only, sequence tumor. That's fine. Okay, everything is good, and I'm just going to submit. Okay, well. So just to give you a quick glance of the mutation spectrum for colorectal cancer, again, majority are R175 mutation, and then the second one is R273 hotspot mutation. So it's very similar to the breast cancer. So now, so again, well, it would be good if you can copy this, what you call it, download this diagram and then include that into the PowerPoint as well, so that you can get a sense of each tumor type. What are the mutations that are predominant? What are the p53 mutations that are predominant? Okay, so but for the data set, I straight away go into the download, go into transpose matrix, and then it will then download into Notepad, and I will just copy it. Okay, and then I'm just going to compile it under the same list here. So I'll just copy and paste here. Okay, I can delete the common title, and this is belong to COAD or colorectal adenocarcinoma. Okay, now so now you can do the rest of, well, you can download the rest of the tumors, okay, and compile it in this list. Okay. Okay, once you have finished compiling, you should have an Excel file, something look like this. So you have BRC, you have the breast cancer, you have colorectal, and all the different 11 or 12 tumors in the PanCan-12, all together about 3,700 ID, and also the p53 mutation status. Now the next thing what we are going to do is we are going to classify each of those cases into different p53 mutation class. Okay, so we'll create another column here. Right now, the first thing we want to do is, well, so you have to understand how many different type of mutation that we are going to group into. So I think, okay, for example, all the NAN will be wild type, and all this FS asterisk here, those are all frame shift mutation, and these are what you call it missense mutation and so forth, and these are truncated mutation and also splicing mutation. So now, okay, to start with the classification, first we will identify all the wild type cases. We will just go to the filter here. So we created a filter bar on top here. We will just look at the NAN. We will just select all the NAN cases. Now, once you apply the filter, so all the cases will be all the will be all the one that is designated NAN by the CBio portal. What we are going to do is we are going to name them as p53 wild type. All right. Okay. Now, so that's good. Now, the next, the next thing what we want to do is probably, uh, okay. Now, if you want, is to identify those cases that have multiple mutations. For example, okay. if you scroll down further, those are some cases that are more than one mutation. We probably also wanted to identify them as well. Now, for example, this case here, we have Q192 truncated mutation and also C242F missense mutation as well. So to do that, what I'm going to do is I'm going to copy this whole list here, and then I'm going to paste it here, and I'm going to use the text to column delimitation. All right, you can put comma as your delimiter. Now that, okay, now then you can apply the filter. So now the first column is the first mutation, basically. Now if you have the second column, if there's any entry here, so basically this will be the one that have the second mutation. So what I'm going to do is I'm going to identify those cases that have more than one mutation. And okay, so what I need to do is I'm just going to select everything except for the blank. So now these are all the cases they have multiple mutation. To a certain extent, this one have 3 mutations on p53 altogether. Now, so what I need to do now, I'm going to label this class here as multiple mutation. Okay. So again, I can then copy and paste it here. So now that I can, so after that, I can delete these 2 columns because I no longer need them anymore. Now, the next thing, okay, we want to search would be those, for example, frame shift mutation, which have a FS asterisk here. Okay, FS asterisk. Okay, or you can use this contain, okay, FS asterisk. Okay, so these are all the frame shift mutation. Now note that you also have some of the mutation which we already identified before to have multiple mutation. So what we are going to do is we are going to uncheck this. So now the list become, okay, now the list are all those with frame shift mutation. So I need to label it as FS. Okay, then I'm going to copy that. All right, so that's done. Now, so I think for the rest of the exercise, I'm just going to leave, I'm just going to check on the blanks one, basically, because those that have designated with the p53 mutation class, I'm no longer worried about it. So I'm just worried those that have not been classified. Right, so the next thing I want to look at would be, okay, R175. Now, so now you have a list of, oh, now you have a list of all the R175 here. Okay, then I can have R273 as well. So these are all the R273. So of course you also have some mutation which are splicing variant. So you see these are all the splicing mutation. I will just put it there, designated them as splice. And what type of mutation that we haven't covered, let's look at that. Okay, now at this point here you will see that, okay, mostly the missense mutation and the truncated mutation are not being identified yet, so we will come to that later on because, okay, and oh, we also have some which is the deletion mutation. Okay, so okay, now deletion mutation always contain this, so right. Okay, so here you go. So now we should have most of the mutation class being identified here, basically. Now, so the next thing what I'm going to do is, okay, to identify all the nonsense mutation. So I have to do it separately again. So because it is a little bit tricky. So what I'm going to do now is basically for the nonsense mutation, I'm going to paste it into a separate list here. All right. And then what I'm going to do now is I'm going to separate them by each of the alphabets, because remember, okay, you have the asterisk. Well, for truncating mutation, you always end with an asterisk. But however, in Excel, when you put asterisk, well, it means everything. So, or a wildcard. So you wouldn't be able to search asterisk. For example, if you search asterisk, it would not happen with anything, or if you filter anything with asterisk, it will not come out, because well, asterisk in Excel means as a wildcard. So what I'm going to do now is basically I'm going to uh, delimited all this data into a single alphabet. All right, so I will go into data. Okay, text to column again. So instead of using delimiter, I will use fixed width. So in this case here, you have to mark one alphabet. That's one. It will separate the TP53, and the T will be in a single column. And then here, P will be single column, 53 will be in single column, and so forth. Okay. So when you click finish, so now you can see that all of them are separated into a single column. So what we want to do now is basically we will look at those that have 1, 2, 3, which are basically end up with asterisk. Oh, sorry, no. Oh, one thing I forgot to do. Okay, before. Okay, so let's see. Okay, this is 1, 2, 3, 4. Okay, this one here. Then you can select those that have asterisk. So this will be all the nonsense mutation. So they have all truncated mutation. So remember, always select this with just a blank only. So this will be all the nonsense mutation now. And you also have to notice is that some of the nonsense mutation are in the residue that are less than 100. So it could, some of it would be here as well. Oh, there's nothing here. And then those are mostly here, I guess. So that's 1, 2. Is there any one here? Okay, that's good. So now that okay, so now that if we look at those haven't identified yet, so these are all basically oh, there's one more here. Didn't come out. I see. Okay. So there are some which is actually on here as well. See that we almost missed that. See these are the mutation that are at the residue less than 100. So you only have 2 digit only. So these are also nonsense. We will copy this. Okay, that's good. Now come back to this page here. So now the blank. So when we look at this, so these are now consist of all the what you call it well missense mutation only. All right, except for okay, that's not available. So other than not available, other than that, these are all the so-called missense mutation. You have some duplication, so we won't bother too much about it. All right, so let's see the non-available. So we will label it as not available, and the rest of this would be okay, missense mutation. Now, but before we look at the missense mutation, I think there's another mutation class which will be quite interesting based on the mutation spectrum, would be the R248. I think we want to create another class for it as well. So just for the curiosity, or okay. So these are R248 mutation class. Now, then okay, and after that, the rest of it, these are basically we can label as missense mutation. I'll just makes sense, we do. Okay, I think we have one duplication. Okay, just to double check, oh, we have one duplication here. We just put duplication, which is quite rare. Okay, good. So now that okay, so now that's done, I can delete this column here. So now you can see that each of the cases are now matched with a different p53 mutation type. Okay. Okay, the next thing what we are going to do is we are going to look at what is the frequency and how many cases of R273, R175, and also the nonsense mutation are actually occurred in each type of the tumors. So what we are going to do is we are going to go into a new Excel sheet here. So I'm just going to paste all the cancer here, basically all the 12 different tumor type, and then I'm going to remove duplicates. So it will remove all the duplicates. So basically these are all the tumors that we have, and we want to look at the number of cases that are p53 wild type and R273, R175, NS, and also other mutations. Okay, all right. So, and of course the total. And we will create some of this column here. So now to look at the wild type cases in breast cancer, so I'm just going to select here tumors and select for uh, BRCA. Okay, and then after that I'm going to select this with just the wild type only. Now, so then you get a lot of the wild type cases. So now just to count them, basically I'm going to paste them into a new sheet, and then I can know that, scroll it down, then altogether there are 681 wild type cases. Okay, so I'm going to put in here 681. All right, so I'm going to go back here, and now I want to look at the R273 mutations. How many cases are those? So again, just paste it here. Altogether, that's only 14 cases. And then R175. Okay, slightly more. 75 is 18. Okay, and then you also have the NS as well. So to see how many NS mutation altogether, there is 46. So now there are other mutations. So well, we don't have to select it. What we are going to do is we are going to identify how many total cases of the in the BRCA cohort, and then use that number to minus off whatever mutation that we have identified. So uh, this is how we are going to do it. Basically, oops, sorry. Here you go. So we are going to select all the different mutation type. So okay. So altogether we have about 982 uh, breast cancer uh, cases in our study. Oops, sorry, how many again? 982. Okay, so here you go, 982. So then here would be equals to the total number minus sum of the cases that we know. Okay, so we have 223 cases. Those are of other type of mutations, which is not categorized by our classification here. So the total, we just want to match the total again. So this total at the end should be the same as here. So now what you can do is then you can repeat the same process for the different type of tumors. Okay. So after that, well, then we can then plot the graph to see what is the frequency of the tumors that have different type of mutation in different tumor types. So this is an example of all the cases that have been calculated. Now the next thing what we want to do is we are going to calculate the frequency of the p53 mutation in each separate class. So what we will do here, then we will use back the same heading again. So now to calculate the frequency basically is equals to the number of cases in each of the category over the total number here. All right, so what you will get is, well, this is basically to give you a percentage. You can change that into percentage. So there are 91.9% of the AML cases are actually wild type. So you can actually put a string, a dollar string here, so that it will lock this column. Then you can basically copy and paste across, right? So now that you have this, is over here, R273, just to double check. Okay, so this will then give you the frequency of the different p53 mutation in different tumors. So now, so next we are going to create a graph to showcase all this mutation. So what we will do here is we will copy the data set into a separate area. So we will just paste the value only so that this part here will still remain as the number. So we will change it back to frequency here. So actually we do need the number here. We probably just want the frequency only. All right. So what we are going to do now is basically we are going to plot a graph. So but before we plot a graph of the R273 mutation in different type of tumors. So before we do that, so we are going to sort the percentage or the frequency of the mutation based on R273H. So okay, you select R273, largest to the smallest. Then here we got ovarian cancer have the highest frequency of R273 mutation. Then we are going to plot this graph here by inserting just a simple column graph will do. Okay, you can put it at the side here if you want to resize it. Okay, then you can select the data. So series name, you don't have to worry about it. So you would plot the frequency, and the x-axis would be all the different tumor type. Okay, so here you go. So you can then change the font size to 12. Okay, and change all the font into black color, and then you can also you also need to label them as well. So this would be your, so the simplest would be okay, so oops, sorry, axis title, primary horizontal, this would be, and also axis title, primary vertical. So this will be mutation frequency, and then this will be your tumor type, right? Bold it. Okay, so you look here that, well, the x-axis here, basically they skip one lane because this is Excel. They will automatically uh, skip one level if they are two clamped together, but in our case we actually want to display all the y-axis. So we double click the y-axis here, I mean the x-axis here, then you go to label. So you change this to specify interval unit to 1, so then you will get all the cases being displayed now. Okay. Oh, so we actually don't need the total. We can minus off the total. Okay, here you go. Then if you want, you can change the color into say red color if you want to. Okay, and then the border also red color. Okay, so from here what we can see here is basically the kidney cancer and the AML have the lowest frequency of the R273 mutation. So we will repeat this exercise for individual graph to look at, okay, R175 and also NS. All right, and then after that, you can copy and paste all this graph into the PowerPoint. Now, at the end of this exercise, okay, if you have downloaded all the graphic from the CBio portal, you should have a p53 mutation maps look something like this to look at the mutation spectrum of different type of tumors. Now, in here, what I'm arranging it slightly differently is basically in the first part of the diagram of the 6 tumors here. These are the tumors that have a high level of R273 and R175 mutation. So you have to label this individually in PowerPoint, basically. Now, and then on the second part of the graph here, so these are the cases that have less, well, or the least frequency in terms of R273 and R175 mutation. And of note here is that the AML and also KIRK, so especially KIRK, the mutation spectrum is very different. There are almost no R273 or there's any of the R175 mutation. So this will be, you can rearrange it in a way to ease your discussion. If I would want to put it as that, well, these are basically the cases that we wanted to look at it in the subsequent analysis because they have high level of R273 and R175 mutation, whereas this group of tumor probably something that I'm going to exclude it from our analysis. And to support that idea, basically this is another graph, okay, to show the mutation frequency of different p53 mutation in the different tumors. For example, okay, we plot that for the R273. If you plot the R175 and the NS, that is exactly how it will look like. Now, to get those cases that are needed for the subsequent analysis, basically we need to have at least 3 tumors with designated mutations in every of the single cohort, because most of the statistical tests, including t-tests, you actually need at least 3 samples to actually get a meaningful p-value. That's why any tumors that have less than 3 cases in a designated mutation class, so I'm not going to include it. So basically for the following exercise and from now on, what we will focus on is basically the first 6 type of mutation. So ovarian cancer, colorectal cancer, uterus, okay, lung, squamous cell carcinoma, head and neck. Now BLCA, even though they have high uh, frequency of R273 mutation, but again, if you look at, okay, let's say here, they are actually quite low, and the number size is actually quite small as well. In the whole cohort, it's only 66 tumors, so I'm going to skip that. So GBM, the same thing as well. So you have no nonsense mutation, so we are going to exclude that as well. Now, of course, you have BRCA, which have, okay, uh, both which have the mutation at R273. Okay, you also have R175 and also in nonsense mutation as well. And the other remaining 3, the lung adenocarcinoma, AML, and also KIRK, this will be excluded as well because they don't have enough cases to make a meaningful analysis in terms of the tumor type specific p53 mutations. Okay, so far we have looked at the p53 mutation spectrum for all the pan-KRAS tumors, and we also look at the specific p53 mutations, okay, in the individual tumors as well. So we are not going to look at the wild-type p53 and p53 mutants expression at this point. Instead, what we will do is we will look into straight away into the clinical outcomes of individual mutations. Okay, in this exercise, okay, we will continue on from the previous Excel sheet, which we already identified each of the case ID that are associated with different type of p53 mutations, and we also designated the different class of p53 mutation as according to the need of this study. Now, what we are going to do now is basically to input in the survival data. So to do that, we are going into cBio portal again to download the survival or the clinical outcome data set. So, okay, now for example, we will in this exercise here we will use breast cancer here. Select that again. We only want to look at mutation and make sure it's sequence tumors, and you put in TP53 here. Okay, you submit. So in fact, this is also quite familiar and also quite the same as what you have done in the previous exercise as well. For example, we have done the similar for mutations to look at the mutation spectrum here. Now in this case, well, you can actually do the same exercise together if you want to. Now, but in this case here, we will be interested in just the survival data set. If you click on the survival tab here, you can straight away get the graph which is plotted by the software. Um, to look at, okay, the survivor that are associated with p53 alteration or without alteration, but we won't be using this graph here. Instead, what we will do is we will download the raw data set here. Okay, so it will download in the text format. All you need to do is you just uh, copy the whole data set and then you paste it here. All right, so you have all the case ID here, and then the status, and also the times of the survival, how long it is. All right, so we can delete this, and then if you scroll it down, so now when you download the raw data, so it will actually separate into two parts. One is the cases with alteration, another one is cases without alterations. But in our case, we just want the raw data. We don't actually need to separate them just yet. So, okay, and then after that, we will basically these are all BRCA to indicate that these are all the BRCA cases. Okay, so now so we will label this as overall survival. You can also create another tab which is for the disease-free survival at the bottom here. Again, you do the same. Okay, you download all the data. Okay, and then you can put it here. So this is going to be all BRCA cases. You can label them. Okay, so that's good. Now the next thing is that basically you will repeat the same process for the rest of the tumor types. Okay, for example, here I will show you another example which is on colorectal cancer. Right, and submit. Okay, straight away you can go to the survival curve. Okay, then you download the overall survival data. And you straight away paste, cut and paste it here. And this will be colorectal cancer. Okay, so now then you can then repeat this whole process to download all the 11 or the 12 tumors that are in the PanCan 12. Okay, once you finish downloaded all the survival data, you should have an Excel sheet look something similar like this. So with all the tumors ID and also tumor types, and also with all the survival data set. Now, one thing I want you to note is basically look at the case ID here. They are okay. They only have three barcodes here, TCGA, AA, and then followed by the patient's number. Now, compared to your case ID, so which is quite different, you have four sections of the barcode. So, including the first three, which is similar to the survivor data, and then you have another extra section, okay, which is not indicated in the survivor data set. So what we are going to do now is basically we are going to okay, input all the survivor data into the same data sheet here to basically compile all the data so that we can later on plot a survival curve. Now to do that, the first thing I need to do is to match the case ID. So I'm going to create another column here, basically. All right, and I'm going to copy all the case ID from here. Now, if you don't want to mess this around, so what you can do is you can create another uh, extra Excel sheets, create another copy here. So I will just put it there as summary because we are going to compile all the data into this summary sheet here later on. So, right, I'm going to copy all the ID. So, paste it here in this column. Now, the next thing what I'm going to do is I'm going to separate, okay, each section of the barcode into individual column. So again, we can do this quite conveniently using the text to column separation. So okay, we can separate by a dash. That means every time when they encounter a dash, it will separate into a different section, right? So you will get something like this. Now, remember, okay, the overall survival and all the survival data, they only have 3 sections of the barcode. So actually, you have these 3 sections that's needed, and this part I don't actually need it. So I'm going to delete the information here. I'm going to insert a column here, basically to put back in the dash. All right, I can copy this and then copy this as well. Okay, so the next thing what I'm going to do is I'm going to combine all this back into the barcode similar to this as well. Okay, concatenate. Okay, 1, 2, 3, 4, 5. All right, so now that you have the barcode which only have 3 sections, so I'm going to copy this and then paste it here as value only. Okay, and then the rest of the column that we can actually delete it. So now that you have the case ID which we downloaded in the early exercise to look at p53 mutation, which consists of 4 sections, and also the ID which is similar but only with 3 sections to match with the case ID of the survival data set. So next what I'm going to do is I'm going to create another 2 more columns. This will be OS and status and also time, which is months. So okay, the same also for disease-free survival. Now, to match all the data into into this based on their ID, I can use the command which is called the VLOOKUP quite conveniently. Basically, if you are not sure, you can Google how the VLOOKUP in Excel works. Here, basically, you just need to put in the formula VLOOKUP. So I'm going to put, see, it's straight away the lookup value will be this one. And then next, I'm going to select the table array, which is actually the whole array here in the overall survival curve. So I'm going to lock it, and then I'm going to, okay, now the column, okay, the column index. So basically what I want to match is basically each of the case ID. I want to know the status here. So which is, this is column number 1, 2, 3. Okay, so for status, I need them to match with the ID and then return the results from column number 3. And then the range, it's only have 2, basically. Okay, so if you put false, basically it will return everything, okay, that is a perfect match, right? So now that's okay, that's how we get it. Oh, so one thing that is missing is this one here. Okay, see, now that you can tell, for example, okay, this TCGA AR A1A, are they, just to double check and search whether it's the subject disease. Yep, so it is disease. All right. So and then the next thing I want to do the same, but to match at the time and the months of the survival, basically the data. I'm going to do the same. So I'm going to make it up here. Now the column of the data that I wanted to return is no longer column number 3, because this is column 1, 2, 3, 4, 5. I wanted to return the data from column number 5. Okay, so this is, so the month is 1721. Again, we look at it. So is that correct? So let's see. Let's check on it and see whether it is correct. See that 1721 disease. Okay, so it's correct. Now what I'm going to do now is I'm just going to copy the whole uh, formula into here. So now that you have, okay, the whole data set that are matched to each of the individual ID with all the survival data set here. So you will basically do the same for disease-free survival as well. So okay, after you have finished compiling the whole data set, you will have something look similar like this. So you have all the different case ID that with all the different disease status and also the time as well. This is a full data set. Now the next thing what I'm going to do is I'm going to replace this non-available data destination here with just a blank, basically. I'm going to replace this one with a blank, replace all. Alright, so now you have deleted all the uh, hash NA cases. Now the next thing, okay, what we are going to do is we are going to select the tumor types that we are interested to analyze. Remember in the previous analysis, we only decided to look at, okay, 6 different tumors. So they are the BRCA1, colorectal cancer, head and neck, okay, lung squamous cell carcinoma, ovarian cancer, and the endometrial cancer. So these are the 6 tumors that have enough p53 mutation cases for us to do further analysis later on. So I will just select these 6 cases here. All right. Okay. So now, oh, but before okay, now what you have here is that under the status, okay, so you have disease or censored. Now, when we input this data into the Prism graph pad, unfortunately, they won't be able to recognize this. So what we are going to do now is basically we are going to change this into the format that they recognize. For sensor cases, basically, you would designate it as a 0. All right, and then for disease cases, which we will designate it to 1. All right, so okay. Now the same for the disease-free survival. So the sensor cases is always 0, basically. Okay, and then uh, you have, which is the case or the event cases, or in this case it will be relapse, will be all designated to 1. Okay, good. So now we have the full data set here. The next thing what I'm going to do is I'm going to import all this data into the graph pad to or Prism to actually plot the survival curve. Okay, select the survival curve here, press create. Okay, so I'm going to label the axis here, which is the x-axis will be time, which is month, and then you have wild type p53, R273, R175, and NS. So, okay. Now, what I'm going to do here is basically I'm going to import the data into GraphPad, but before that, you note that some of the cases there's a missing data point, basically. So those are quite common in most of the clinical study. So what I'm going to do is I'm going to exclude all the blanks, okay, filter it out. So now you have all the cases that have survival data set. I'm going to copy the case ID. Oh, before I do that, I need to select the cases. So first I'm going to look at the wild type cases. Alright, so this includes all the 6 tumors that we are interested to look at. I'm going to copy and paste the title here. Alright, then after that the status, oh, sorry, the time first. Then, okay, so then the x-axis will be the time, and then the y-axis here would be all the 1 and 0. Okay, so the 1 is disease and the 0 are the sensor cases. All right, so that's good. You scroll down to the end. Now, then you repeat the same process. Basically, now we are going to change this to R273 only, and you will select the cases. Okay, then you are going to select the time. Then you will the status. Now the status, make sure this time here you are actually input it into the R273 column. All right. So then you would do the same for R175. Again, cases. Then the time. Status. Okay, now this one here will be R175. See that? Okay, and the last column would be the NS. Okay, when you have plotted all this, so then you should be able to see a graph something like this. All right. Okay, so we do need the title here actually. We can delete the title. Now we can adjust the scale a little bit. So to make it basically 120%, that would be good so that you can see a graph, something like this. Okay, now the next thing you can do is that then you can label the color of the individual bar. For example, wild type, we will stick it to black color. R273, so we will basically we will change it into say red color. Okay, and then for R175, we will do it as the green color. And for the NS, we will then change it into blue color. Okay, then you will get a survival curve like this. Now, if you go to curve comparison here, it will tell you basically a p-value based on the log rank test. So here what it shows is that the p-value is set at 0.001, which is quite significantly different. Now, however, if you plot all the four graph over here, the p-value here is just tell you that the curve would be of the individual curve are significantly different, but it doesn't tell you whether it's significantly different between the R273 versus the wild type, or between the R273 versus 175 and so forth. So this is a group analysis. So what I'm going to do next is I'm going to compare it individually to a pairwise analysis. Basically, what we will be interested in will be, for example, okay, the R273 mutated tumors, are they significantly different compared to the wild type in terms of their survival? So to do that, okay, I'm going to uh, go back to the data sheet here. So just delete the two columns with the data R175 and the NS. So you can delete it, and then now that it only show two curve here. When you go to curve comparison, it will tell you the p-value, which is 0.002, which reflects basically the p-value by comparing the R273 versus the wild type. So I'm going to undo. Okay, that's how you can get the p-value by doing a pair comparison. So if I undo it, okay, and then now if I want to compare it to R175, I'm just going to delete the R273 and also NS. Then now you have the graph here, and then the curve comparison will give you the p-value is equal to 0.0123 and so forth. Okay, you can repeat this whole process for both the NS, I mean for both the disease-free survival and also overall survival. Okay, once you finish the exercise, you should be able to get a survival curve, something like this, basically. Now, what is quite interesting here is that by looking at the disease-free survival, okay, so there are significant difference between the R273, R175, and also uh, NS mutation compared to the wild type. Now, the p-value you can decide to call it a significance, whether it's p less than 0.05 or less than 0.01. So in this case here, I think we will make the statistical significance will be defined as p less than 0.05. So okay, this is for disease-free survival, and this is for overall survival. Again, you will see that okay, they are quite significantly different compared to the wild type as well. So based on this data, what we can conclude is basically the R273, R175, or the nonsense mutation are basically have a poorer survival outcomes compared to the wild type p53. Now, this concludes the whole part one of the analysis, which is mainly just to give idea about the p53 mutation in the PanCan 12 dataset.